today I'm going to be discussing the cell cycle and how it is controlled by internal and external mechanisms. Also, towards the end of the video, I'll go over how the loss of this control can lead to various problems. The cell cycle is a highly regulated and controlled complex set of stages. These stages include interphase, which is indicated by the I in this diagram, mitosis, which is indicated by the M in this diagram, and cytokinesis, which actually just happens at the end of mitosis. The cell cycle is essentially mitosis, alternating with a much longer stage called interphase. In fact, interphase actually accounts for about 90% of the cell cycle, which is easily shown by the orange in this diagram because it leaves just a little tiny bit of a portion for mitosis while it takes up most of the cycle. During interphase, a cell grows and copies its chromosomes in preparation for cell division. It can also be divided into three subphases, G1, S, and G2, which I'll go more into depth about in just a moment. However, I would like to mention that after interphase, the cell enters mitosis, which is the division of the nucleus. Mitosis can also be broken down into different stages. Right over here it is shown. This is actually interphase because you can see that the chromosomes are not replicated. But right here, mitosis starts with prophase, metaphase, which I'll also describe more in detail later, anaphase, and telophase, and also cytokinesis if it goes through that. And cytokinesis is a division of the cytoplasm and other cell components which completes mitosis. As mentioned before, interphase can be broken down into three subphases, G1, S, and G2. G1, also called the first gap, is where the cell grows. S is actually the second gap, and it is where DNA replication occurs, which is obviously very important. And G2 is quite similar to G1 because it is also when the cell grows in preparation for cell division. Also, I talked a little bit about how mitosis has different stages to it, which is more described in this picture. Before mitosis, the interphase right over here happens where chromosomes duplicate. However, this is actually in the form of DNA is actually in the form of chromatin in this stage, and in the next phase, which is prophase, which is actually the first stage of mitosis, the chromatin condenses into a chromosome form, which is a much more compact form of holding the DNA. And not only does the chromatin condense into chromosomes in this phase, but the nuclear envelope also starts to break down, as you can tell. And you can also see that the spindle fiber forms, which actually plays a very important role in mitosis. Next up is, I did not mention this, but this is the prometaphase, which a lot of people seem to look over, but this is also important because this is when the nucleus membrane breaks down, and then you can see in metaphase it is completely gone. And during metaphase, the chromosomes line up along the middle of the cell, which is also called the metaphase plate, like on the metaphase plate. And then moving on to anaphase is when the sister chromatids actually begin to separate and pull apart from each other, and telophase is when the two groups of chromosomes reach the opposite ends or poles of the cells. Now there you can see the two cells are pretty apparent. Trying to circle, but it's way off. And then if cytokinesis does occur, which again is the division of the cytoplasm and the cell organelles, the result will be two genetically identical diploid daughter cells. Like I said, they're diploid, and not only are they identical to each other, but they're genetically identical to the beginning parent cell right over here. So these three cells are genetically the same. Okay, now that we have a better understanding of the cell cycle and its components like interphase, mitosis, and cytokinesis, we're going to move on into the checkpoints of the cell cycle. A checkpoint is a control point where stop and go ahead signals can regulate the cycle. These help guide the cell through the cell cycle. I mentioned how stop and go signs are given at each cell cycle checkpoint. However, why are checkpoints so essential to the cell cycle? This is because they ensure that all of the important cellular processes that should have happened until that point have happened correctly, and therefore will tell whether or not the cell the cell should continue with the cell cycle. There are three major checkpoints on the cell cycle. The G1 checkpoint, which is right over here. The G2 checkpoint is right over here. And the M checkpoint, which is right over here. Cells pass the G1 checkpoint, which is located right over here on this diagram, between the S and G1 phase when they are stimulated by growth factors, cell size, or the environment. Remember, growth factors are proteins that are released by specific cells that encourage other cells to divide. If the cell does not receive a go-ahead signal at this checkpoint, at this initial checkpoint, it will actually switch into a non-dividing state called G0, where no dividing will occur. Moving on to the G2 checkpoint, you go past the S cell phase, G2, and then you get to the G2 checkpoint. Here, which is located from the G2 to M transition, this checkpoint ensures that there is no damage after DNA replication, and if there is damage, it does not allow the cell from continuing the cell cycle into mitosis. Lastly, the M checkpoint, or the M spindle checkpoint, checks that the mitotic spindles or microtubules are attached to kinetochores, which are structures of proteins that are attached to the centromere, which is located right over here. These are the kinetochores, and which are attached to the centromere that links each sister chromatid to the mitotic spindle. If they are not attached right, the cell cannot continue to mitosis. Like I said earlier, cell division is a highly regulated process. It is tightly controlled by complexes that have enzymes which are called CDKs, or cyclin-dependent kinases. They are called CDKs because it is composed of a kinase that must be attached to a cyclin right over here in order to perform. So what do these CDKs do? They turn on or off the various events that occur in cell division. When the CDK is bound to the cyclin, which is shown right over here, it produces an activated complex that interacts with other proteins that it allows the cell to go into mitosis from the G2 phase. The CDK and cyclin are like switches for the cell cycle, and it will help the cell to progress through the cell cycle. On the CDK is a thing called mitosis promoting factor, MPF for short, which contains cyclin A or B attached to CDK. Over here is just a cyclin graph that shows different concentrations of the cyclins as you go through the cell cycle. You can see cyclin D kind of increases in concentration during G1 phase, and then just kind of levels, and then at mitosis it decreases. Cyclin E, it increases in between the G1 phase and then the S phase. Cyclin A, it starts to increase, especially in the G2 phase, and then starts to decrease towards mitosis. Cyclin B increases until it reaches this checkpoint right over here, and then it decreases while it goes into mitosis. Like I said, different CDKs are produced during different times of the cell cycle. As each cyclin turns on or off, CDK moves the cell along the stages of the cell cycle. I mentioned how stop and go signs are given at each cell cycle point. However, what provides these stop and go signs? Internal and external signals. Many external factors, including chemical and physical factors, can both influence cell division. An example is a growth factor, which I said earlier is a protein released by specific cells that encourages other cells to divide, is a platelet-derived growth factor, or PDGF in short. Right here is a picture of a sample of tissue from a human being that is cut up into pieces. PDGF is added to half of the culture vessels, which is shown right over here, and not added into the other half. And as you can tell, the growth medium with PDGF caused cells to divide in increasingly amounts, showing all of these cells that divided, and then in the one without it, the cells fail to divide. And the PDGF is needed for the division of fibroblasts, which is a type of kinetic tissue cell, which is shown right over here in this picture. The fibroblasts have PDGF receptors, and when the PDGF molecules bind to them, it triggers a signal transduction pathway that allows the cells to pass the G1 checkpoint and allow mitosis to happen. What happens when something goes wrong with the cell cycle genes and it interferes with the cell cycle control? Cancerous cells are produced, unfortunately. Cancerous cells do not respond normally to the body's control. They may go on dividing despite overcrowding or detachment, and also they may not need growth factors to grow and divide, or might just produce their own growth factors. This diagram shows normal cells versus cancer cells. As you can tell, these are way more dysfunctional, and they're just, the nuclei is way bigger. And then here it shows that these dangerous cells can bunch up and cause a tumor, and these are the normal cells versus the cancerous cells. In conclusion, the cell cycle is mainly composed of phases including interphase, mitosis, and cytokinesis. Also, the cell cycle is highly regulated by checkpoints. The three major ones are G1, G2, and M. Internal and external signals give the stop-and-go signs at these checkpoints. Cyclins and CDKs are extremely necessary for the cell cycle to be successful. If there are disruptions in the control of the cell cycle, unfortunate outcomes can occur like cancer. That is all for this video, and I hope that you enjoyed these cartoons after a very serious discussion, and I hope that this video was very helpful.